Hello, everyone. My name is Sarah Jane Demy, and I am the founder and CEO of Demy-Colton and located here in New York City. It is my pleasure to welcome you to our 16th installment of the Demy-Colton Virtual Salon series, Targeting mRNA, the New Frontier of Tailored Therapeutics, and it will be a lively discussion. So thank you all for joining us. As some of you know, Demy-Colton's mission is all about convening biopharma and digital medicine industry leaders to catalyze change. We know we help drive innovation when we bring individuals together who are united by a common cause and a shared passion. And in our industry, that cause and passion is bringing therapeutics and cures to people in need. As those of you who have attended past virtual salons know, Demy-Colton was in the process of launching BioFuture, our new international investor and business development conference here in New York City, when COVID-19 changed everything about our world as it did about yours. Therefore, in lieu of an in-person BioFuture meeting, we've launched the Demy-Colton Virtual Salon series to continue our mission of bringing people together with great and informative content. So before we get underway today, I would like to acknowledge and thank our WeHi, a diamond sponsor of our virtual salon series, as well as our founding BioFuture sponsors for their ongoing support. At Demy-Colton, we believe the virtual salon series will be here to stay even after we get back to a new normal, whatever that new normal is. So please do check our website for a schedule of salons, fireside chats, topics, and participants. It is evolving constantly, and of course, I encourage you to suggest topics and to join us at future events. A note about the structure of today's program. The formal portion of the discussion will last approximately 50 minutes and will be followed by a 15-minute Q&A period. You can submit questions in the Ask a Question pane below the screen, and we will address as many as possible. And of course, I encourage you all to make your questions as concise as possible so we can get to as many as possible. Please note that this salon will be recorded and will be available for on-demand viewing today. Additional materials can be viewed on the Event Resources tab. Today's virtual salon, Targeting mRNA, the New Frontier of Tailored Therapeutics, will be moderated by Lyndon Pullen, founder of Pullen Consulting and a long friend of Demy-Colton. And she is joined by our panelists, Robert Copeland, President, Co-founder, and Chief Scientific Officer at Ascent Therapeutics, Joan Heath, Associate Professor at WeHi, Carolyn Ng, Managing Director, Vertex Ventures, HC, Jennifer Fetter, Founder and CSO, Arrakis Therapeutics, and last but not least, David Siegel, Senior Business Development Manager at WeHi. I welcome all of you. We are pleased that you could all join us here today for what we know will be a really great discussion. Now, full bios of all of our panelists are available on our website. And without further ado, I'd like to turn the virtual microphone podium over to Linda. Linda, the stage is yours.
Thank you, Sarah. I'm delighted to be here when the Walter and Eliza Hall Institute, Australia's premier research institute, asked to sort of pull together a panel on this topic. I thought it was a great topic. How often do we get to talk about such a hot topic, thanks to the awareness that COVID vaccines have given to mRNA, but also one that really is a frontier? When we think about the past history of drug discovery, we targeted proteins, we've thought about genes and gene therapy, but the majority of us have not thought about targeting mRNA, the messenger RNA that arises from DNA to RNA to protein. And that's what we're going to talk about today. I've got a stellar panel representing diverse backgrounds, lots of industry experience. And so with no further ado, I'll jump in and ask each of them to introduce themselves. I guess we'll start with you, Bob Copeland, alphabetical or something like that. Would you please give a brief background and how you tie into this field?
Thanks very much, Linda. I'm the Chief Scientific Officer, co-founder, and president of Accent Therapeutics, where we're focused on developing small molecule therapeutics that intervene in RNA biology by inhibiting the enzymes that modify RNA, and in that fashion, control all aspects of RNA biology. I'm a chemist by training, enzymologist by reputation, and have been doing drug discovery in large pharmaceutical and biotech companies for a little over 30 years.
Thank you. Joan?
Yes. Hello, I'm Joan Heath. I'm an associate professor and laboratory head at the WEHI in Melbourne. Some background about our research. Well, my lab undertook an unbiased forward genetic screen in zebrafish with the aim of identifying genes that are required to maintain a highly proliferative cell state. And much to our surprise, of the genes we identified, many encoded proteins that are involved in RNA processing. And we've spent several years evaluating whether these proteins might be worth targeting for the purpose of cancer therapy. And we're very excited about the potential of small molecules to target RNA molecules directly. And so I have to say, I'm greatly looking forward to hearing about the drug discovery efforts of my fellow panelists today.
Thank you, Joan. Jennifer.
Hi, I'm Jennifer Fetter. I'm the founder and CSO for Arrakis Therapeutics. I'm a chemist by training, and I've worked at a number of large pharma and biotech companies over the past, well, I have to admit, several decades. Arrakis is focused on interdicting messenger RNA and pre-mRNA small molecules, drug-like small molecules that contact directly folded RNA structures that dictate the function.
Thank you. Carolyn.
Hi, everyone. I'm Carolyn Ng, Managing Director here at Vertex Ventures HC. So our fund is actually a Singapore-headquartered fund backed by Singapore's Temasek Holdings, but investing globally in early-stage biotech and medical devices companies. So we typically invest in early-stage Series A, Series B companies and have done a lot of oncology and various therapeutic areas. mRNA, RNA therapeutics as a whole remains a strong area of focus or interest, I would say, for us. And we are also investors in a company called 27, where I'm on the board of, where they are discovering and developing small molecules to target RNA modifying proteins. And my scientific training was also oncology, molecular oncology specifically, and then was in pharma consulting for a number of years, working with global pharma such as Takeda, Roche, and Pfizer. Great to meet all of you today.
Thank you. David.
Yep, thanks, Linda. Hi, everyone. I'm David. I'm a Senior Business Development Manager here at the Walter and Eliza Hall Institute, which we call WEHI. Before joining the commercialisation team at WEHI, I had about 15 years of working on commercially focused drug discovery and target validation work, both in academic settings and also in industry. And that work was focused mostly on cancer and metabolic disorders. At the moment, my role is to manage the translation of the discovery science of cancer assets here at WEHI.
Thank you. So let's kick off with some questions. And I'll start off with Jennifer. What is mRNA and why target?
So, all right. So mRNA, in the central dogma, DNA makes RNA makes protein. And so mRNA is the message in which we transcribe nucleic acids off DNA. And then those, after some editing and processing, that RNA is exported to the cytosol. The ribosomes decode it, in essence, and translate it into protein. And so that's basically what it is. The reason to target this is that it's upstream of all of the proteins. Proteins in some sense, are just the henchmen. If you want to go after, the bosses, RNAs represent an enormously complex regulatory apparatus that guides much of what happens in cells. And I think we'd like to intervene in that biology and develop molecules to allow us to do that.
Thank you. And so what's different, Joan, about targeting mRNA from targeting proteins or genes themselves?
Well, I just think, following on from what Jennifer's just said, on a conceptual level, targeting mRNA presents you with the opportunity to shut down the expression of an unwanted protein, let's say, at an earlier stage in the gene expression cascade than targeting a protein. And that might be quite advantageous. It might be a more efficient or effective way of doing things. But there are many similarities, I would have thought, as well as differences. Ultimately, you're trying to identify small molecules that fit precisely into features that are either unique to the targeted mRNA or its encoded protein. So I would have thought, the need to design good chemical screening strategies would be the same for trying to identify molecules that interact with RNA as you would design for identifying small molecules that interact with protein. I guess the big question, we know, for instance, if we find a small molecule that's targeting a protein effectively, it's likely to be fitting very precisely into a catalytic site or a discrete groove on the protein surface. And if you find such a small molecule, chances are that that interaction will be limited to that protein and perhaps some very closely related family members. I guess the big question for mRNA targeting is whether we're going to be able to have strategies that make it possible to target individual mRNA selectively. I think if that became a reality, that would be absolutely amazing. But I think at this point in time, it's not possible to say whether we'll be able to get that level of selectivity.
Comments on that, Jennifer? You look like you're thinking about that one.
Yeah, we've actually found selectivity to be fairly straightforward. So much as Dr. Heath was saying, we actually do screen using protocols that are at least roughly analogous to the screening methods you would use for proteins. And selectivity is, of course, something that we're concerned about. And we do see some overlap, molecules hitting more than one. But actually, when you do a typical set of screens of the number of RNAs, it's maybe the kind of selectivity problem you would expect to see by screening a set of kinases. There'll be some overlap, but plenty of molecules highly selective. So I actually don't think this is where the real problem is. And I should say, this is not based on 2 or 3 screens. This is based on about 30.
That's wonderful. So, Robert, are there differences in targeting splicing versus translation or other sorts of roles on your mRNA?
Well, I think that there are some differences in those processes, but importantly, I think there are also some very common features. Splicing occurs at the spliceosome, and translation occurs at the ribosome. And both of those are aggregates of RNA and protein that form hydrophobic pockets that are well adapted to small molecule drug-like interactions. As a matter of fact, one of the programs that I worked on when I was at GSK targeted the bacterial ribosome over the mammalian ribosome. And that led to what is today an approved antibiotic, Altabax. So I think that there's plenty of opportunities for targeting splicing, targeting messenger RNA directly, but also there's tremendous opportunity to do what we're doing at Accent, which is targeting the enzymes involved in modification and processing of RNA. And those enzymes control every aspect of RNA biology that one can think of. And by targeting the enzymes, we're taking advantage of over a century of medicinal chemistry know-how in how to target small molecules to enzymes and develop therapeutics that are orally bioavailable, that have appropriate pharmacokinetics for convenient dose levels and dose scheduling. And so, by and large, we think that the intersection of 100 years of enzyme-targeted medicinal chemistry with the emerging exciting area of biology of RNA modification and RNA processing is a real sweet spot for us.
Carolyn, is 28.7 focusing on the mRNA itself or on proteins associated with mRNA?
So it's actually also proteins that are associated with modifying or degrading or processing RNA. Yeah. And in fact, actually, as the name suggests, it's 28.7. So where the initial work, the company was founded on George Daly's lab and Frank Slack's lab in Harvard, where they identified Lin28 as being a protein that modifies the levels of lead-7, which is a microRNA implicated in cancer. Yeah.
Right.
I was just going to ask, if I may,
Sure.
Bob, whether there are any implications for the fact that obviously splicing takes place in the nucleus versus translation in the cytoplasm. Are there any issues with small molecules getting into the nucleus, or do they just diffuse in easily?
So there doesn't seem to be any major barrier to that. There's passive diffusion. There's also active transport of molecules, metabolites, and things of that sort into the nucleus. And a lot of small molecule drugs that act on RNA polymerases, DNA polymerases, various kinases that are associated with the nucleus, kinesins that act as centrosomes. All of these seem to work fairly well. So I don't see a major barrier there, but of course it's going to be dependent on the details of the small molecule.
That's an interesting reminder of sort of the continuum of our learning from one place to another. That's useful. So I'd love to hear about the challenges, and I'll kick it off to Joan, but everybody please chime in. What are the big challenges that you see?
Well, I guess I'm obviously not very experienced in terms of identifying small molecules that interact directly with mRNA. But I guess my overarching question is, what do we know about the mechanism of action when a small molecules bind to mRNA? I'm sure there are lots of different strategies. But, is the aim generally, or would one of the strongest goals be to try and target individual mRNAs selectively, and then have a mechanism for triggering the degradation of that mRNA? Is that one of the major goals of the field? Because obviously that would be very effective and probably one of the safest things to aspire to. But I can envisage, other scenarios that might kick in where, for instance, blocking some function of the mRNA caused it to, accumulate in its improperly spliced, non-translatable form. And sometimes those types of mRNAs have the ability to hang around in the nucleus for a long time and potentially cause collisions between the DNA replication, transcription, and splicing machineries, and create widespread replicative stress, for instance, that in turn might trigger DNA damage, cell cycle arrest, and ultimately cell death, which, in many instances might be a very desirable outcome. But I don't know whether this is a common goal for the field, to try and induce cell death in this way and thereby potentially eliminate a cancer cell. So I'd welcome other people's views on whether that's a mechanism of action that people are aiming for and whether they think it's tunable and potentially safe.
Jennifer, I'd love to hear your thoughts on whether you're aiming at apoptosis and cell death, or are you aiming at more discrete signaling things?
Yes. So, I should say that for the most part, we tend to think of this in discrete outcomes. Having said that, there's a wide range of potential mechanical outcomes. So just a few examples. One is that there are ways to alter splicing so that they get intron retention, or that you get premature termination of codon expression. And so that can lead to the mRNA being exported just fine, translated just fine, but ultimately the protein that it would normally produce is simply not produced. Another is that if you can impede translation, because you stabilize the structure that's kind of in the way, what you're likely to produce is frankly a backup ribosomal traffic, so that the polysome becomes crowded. And there are actually endogenous mechanisms that called nonsense-mediated decay, which causes the cell to basically say, this is unproductive translation, it pops up the mRNA, sends all the ribosomes off to do something useful. And that's people talking about the 3' UTR, but there's a variety of mechanisms for causing RNA to be degraded more quickly or less quickly. And so the only possible answer to that question is to appreciate that the heterogeneity of mechanisms that you might interdict is at least as great as the heterogeneity mechanism that you find in proteins. And with that sort of horrifying thought in mind, to tackle a few of these and try to make sense of them and exploit those mechanisms, for therapeutic.
Bob and Carolyn, are you chasing only certain categories of mRNA mechanisms, such as degradation, or are you tackling apoptosis, specific signaling? It's an amazing thing to think about, that there are that many different ways to tackle this. I think that's a superb point.
Yeah. In fact, just maybe taking one step back, I know that we have so far been talking about sort of small molecules against as therapeutic modality here. And we have a great panel actually with people like Bob and Jennifer, who are obviously, and Joan, who are obviously experts in that area. But taking one step back as an investor, we've been really sweeping and looking at the landscape of the RNA therapeutics and how that's evolved, from that sort of 6,000 feet on top. And it's been fascinating, depending on sort of either on the oligo side of things, which is kind of where everything first started, right? The first approved RNA therapeutics are actually in the oligo and also RNAi antisense sort of modalities. And that has also been fascinating, but very much challenged by the delivery problem, where we've been having limitations on only targeting indications that have an effect in the liver. So I think that is sort of one area that we're still expecting to see more technological breakthroughs, to see if we can actually effectively use oligos or antisense or ASO in other part of the body that is not the liver. That's one area. There's also alternative ways of targeting or silencing or re-expressing target proteins. There are approaches such as companies like AppicBio, for example, that are, that have we were not investors in them, by the way, but very interesting company that's trying to silence, an mRNA at the same time, and using microRNA to silence a protein of interest, and also expressing a protein of interest with knocking in a normal mRNA. So that's also a very I think, exciting field of using mRNAs as RNAs, broadly as a therapeutic. And then finally, you have the small molecule realm, right? And then you can sort of split that into two buckets, or that's kind of how my investor hat thinks about it. I think one category is really the interesting companies like Arrakis and Ribozyme and Talk, who are all developing small molecules to directly bind or target these tertiary structures. And then you have companies such as Accent and 28-7 and Storm, which are developing small molecules to target proteins. But these proteins obviously have a modifying effect on the RNA, and that could be degrading, that could be processing, that could be epigenetic modification. So I think the possibilities are really so all-encompassing in the RNA therapeutics field. And I think depending on sort of which area you're looking into, there's different levels of biology versus chemistry risk, but all of them, I think, warrant significant effort to go forward and advance.
Yeah, if I can just
there's
add
also
on to
one
that.
other possibility. Sorry, Rob, you go.
No, go ahead.
Oh, just something fascinating we saw just published just recently, which was that the accumulation of RNAs within cells can trigger, we've known this forever, that the accumulation of RNA, particularly double-stranded RNA from viruses, can trigger antiviral responses. And if we do have missplicing, there's some data coming out that is sort of indicating that, these misfolded or misspliced RNAs can also trigger, in certain contexts, antiviral responses, and this sort of holds the promise of being able to turn cold tumours, immunologically cold tumours, hot, and so that the tumours can attract the immune response and wake up the immune response and have an anti-cancer effect that way. So it's a very nice, how can I say it, the intersection between immuno-oncology, which is of course taken off over the last decade, with this new emerging RNA biology in a fairly unexpected way. I guess looking back, makes sense that this is almost obvious, but of course all good ideas are like that. So it's really nice to see that there might be some sort of real left field sort of applications for the work that we do.
I think that's well said, David. Caroline nicely summarized a lot of the exciting things that are happening in the field. I think those are exciting. I think it's going to take time to really sort out a lot of the challenges in terms of, can you get not only the selectivity but the potency that one needs for reasonable human doses by targeting RNA directly? Can you deliver molecules in a convenient fashion to patients? These are all, technical challenges that I'm sure the field will overcome. We've taken a different approach at Accent to say that, if we target enzymes, we know how to solve pharmacokinetic problems. We know how to create molecules that are orally bioavailable. We have 100 years of precedent in medicinal chemistry. So we've chosen to focus on those enzymes involved in RNA modification. When we look, there's over 500 genes that are involved in one way or another in modification of RNA. So this is a very rich target space. It's a very important area of biology. Biology doesn't invest that type of bioenergetics in a thoughtless way. Sorry to be anthropomorphizing, but we think that this is a very rich area. David, the point that you made about the innate immune response and the association of that with immuno-oncology is something that we are very aware of. One of our targets that we talked about publicly is an enzyme called ADAR1, which some very nice work at the Dana-Farber Nick Haning's lab demonstrated that when that is knocked out in vivo, it creates a situation where cancers that were cold to checkpoint inhibitors now become hot, and cancers that were not only refractory but were became resistant to checkpoint inhibitors are now resensitized. And so, we see a lot of tremendous potential value for patients in targeting these type of enzymes.
Thank you. So, Jennifer, I know you're a small molecule drug hunter, but what are your key tools for targeting mRNA?
Well, first of all, you have to find the target. So, you can identify a gene that's associated, that produces a transcript, which is associated with some protein product that's of very high value. So we can just pick one, call it MYC, right? So you say, oh, well, MYC should go down. We think that's a good idea. So, but MYC has been very difficult to drug. There's been interesting recent progress on that protein, but I still think it's a tough slog. And my feeling was it's got to be easier to drug that darn than it is to drug the MYC. So what you have to do is go for the in the transcript. It turns out that about 85% of RNA, just as global average, is highly with varying levels of dynamics. So that's sort of preamble tells us about the kind of tools. So we have to use informatics and chemical probing techniques to make sure we understand where the structures are and how persistent they are inside the cell. We have to couple that with basic biological knowledge to overlap structures with that function. Then we have to build that particular piece of RNA and demonstrate that the structure that we built mirrors the structure that is embedded in the larger transcript. Then we screen it, find small molecules that bind to it. And then the exciting part is to figure out if the molecules have any impact on that RNA intrinsically, and then, more interestingly, in the cells, interdicting that particular structure inside the cell, and therefore it's. And so most of the tools you would find to be fairly similar. Maybe a few that are more important to us because we have to take into consideration the very explicit, the dynamics of RNA to appreciate that. And maybe we do it at a astounding amount of sequencing.
I bet you do, yes. Other comments on the dynamic component of mRNA?
Yeah. So, one of the problems with textbooks is that quite often they're wrong, and the RNA is always drawn as a straight line with a poly A tail. And of course, and Jennifer would know this more than everybody, that of course it's a globular structure and it folds according to thermodynamic principles. And I guess the rate of transcription might impact how the shape forms. But that's not my. It is. That RNA doesn't exist in a vacuum. It's almost always bound to proteins at some point, either at the 3' UTR or in the 5' UTR, and these things can change their shape too. And I'm just sort of wondering, do we know enough about RNA binding proteins to say how RNA is going to look inside a cell? In a particular type of cell, like a highly expressing MYC cancer, or just a regular, a regular cell like a muscle cell, which I mean, do they look the same? Is there an opportunity for therapeutic window there? Lots of questions. I can offer
a view on that, right? So our SAP at the outset had RNA structure. RNA pretty much brings its own structure to the party. And then later to the party are the proteins.
Okay.
And for the most part, our wet lab work has supported that generalization. That is to say, when you do chemical probing experiments on isolated RNA and chemical probing experiments on those RNAs in situ in the cells, you can see there are occasional exceptions. You can see very high fidelity. Moreover, most RBPs don't bind very tightly or persistently. And that one of the points of evidence for that is that when you do chemical probing experiments, you don't see a lot of masking or ghosting. Not to say that they're not there, but they don't seem to sort of set off. So our view at this point based on our data, is that most RVP, I've heard their high estimates 2,500, but something like 1,800 RVP. They will very strongly influence the dynamic of that RNA and perhaps where it's routed. But they don't tend to alter the shape very much, except the helicase. I mean, helicase doesn't find it, but for the most part, they're not controlling the conformation based upon what we see.
That's a fascinating observation. Bob?
It's great.
I'm sorry, Linda, I didn't hear the question.
Well, I was just wondering whether you want to comment on that concept that the proteins are not altering the shape that much of the mRNA.
Well, yeah, I don't have direct data on that like Jennifer does. I'm not going to challenge her work, but what I would say is that by targeting enzymes, we know they're doing mechanisms. And so they're changing the dynamics of RNA. Whether or not they change the structure, they're certainly changing the repertoire of binding proteins that the RNA will interact with. And so there is a dynamic component there, whether it's dynamics of RNA structure per se, or dynamics of localization, dynamics of binding partners. I think that that's still an area of biology that is being unveiled. But what we can say with great certainty, though, is that these enzymes that we are targeting do play a significant role in changing RNA dynamics. And we can see that with knockout experiments. We're starting to see that with small molecule inhibitors at this point.
Thank you. Joan, that's a nice lead into you thinking about model organisms and different systems to study the biology.
Yes, well, I think one big contrast that's evident today is that there are, different reasons for coming to the realization that, messenger RNA is a very important target that, we've all come to appreciate in various different ways. And I certainly think, the way we came about it is quite different from the way people have described today the exquisite biochemistry that they now understand. So as you probably already gathered, we're very fond of zebrafish as a model organism. And, the reason I recommend them is because, in my opinion, they're the best vertebrate organism in which to carry out large-scale forward genetic screens. And the beauty of a forward genetic screen is that they provide a completely unbiased approach to identifying genes that are indispensable for a particular process that you might be interested in. And that unbiased word is the main value of that approach, I think. So in our case, we were interested in understanding the genetic regulation of a discrete stage in zebrafish development when there's a sudden burst in the size of the liver, pancreas, and intestine. And we found that virtually every cell in these developing organs at that particular stage in development was rapidly cycling. And we wanted to understand the genetic regulation of this highly proliferative state, because we thought it was likely to be reiterated in digestive organ cancers too. And of course, they're notoriously difficult cancers to treat. And so we carried out a forward genetic screen. And much to our surprise, we uncovered a collection of genes that in almost every case encoded protein components of massive cellular machineries that carry out a variety of RNA processing reactions, such as transcription machinery, ribosomal RNA processing machinery, splicing machinery, and the complex that carries out cleavage and polyadenylation. Now, this was a complete surprise to us. We were thinking we might find discrete regulators, let's say of the Wnt signaling pathway or something like that. So without this unbiased approach, these genes would never have come to our attention. And we now feel that they're, bona fide great targets for cancer therapy.
That makes me think about the classic dilemma of tumor versus normal and rapidly proliferating tumors versus slowly proliferating tumors.
Well, we feel the context that's most relevant for our work is the oncogene-driven state, when cells are already in a heightened stage of stress. They're still proliferating rapidly, and they're managing to keep their act together. But if you suddenly disrupt splicing or cleavage and polyadenylation, suddenly the stress is too great, and that cascade that I mentioned before of replicative stress, DNA damage, ultimately triggering cell death, that we think is probably an important area that would benefit. Now, a slower dividing tumor, probably it wouldn't be so effective.
Right. Carolyn, can you bring your thinking on how you prioritize different types of mRNAs or different processes, and how you would think about that as an investor as well as a board member of a company?
Yeah, that's probably $1 billion question, to be honest.
Yes.
Yeah, and I think it really depends. Again, I think back to the modality of the RNA therapeutic, as I was describing earlier. Again, if we're not, outside of the small molecule space, if it's really oligos and some other types of modalities using RNA as a drug itself, right? I think those targets have pretty much been specific to the tissue delivery. So I think now we're increasingly seeing targets outside of the liver, perhaps in muscle. I think that is a new area where some work is going into, where there are companies that are developing targets to be knocked down or restored, et cetera, in the muscle with RNA. So if we were to talk about, I guess, back to, I think, where we've been spending a lot of time on this discussion, which is small molecules either directly targeting RNAs or modifying enzymes or proteins that affect these RNAs, then I think the question is twofold. I think depending on whether or not we're starting from a chemistry platform, which I think, Iraq is, like, for example, is doing a fantastic job at, I think and Jennifer will be in a much better position to sort of talk about how they think about target selection. But from the way I see it is probably going to be a combination of several different factors. Is what can we actually target selectively, right, with the required level of potency, and what are those proteins in terms of what do we know or has established of their biology, right, in the right indications. So at the end of the day, it's going to be a combination of being able to hit both and then come up with a drug-like properties that are amenable to drug development. So that's going to be one area of going at it. Whereas there are some other companies, and again, Exxon and the other companies will be in that bucket where they're probably going from the biology standpoint, where we are going for specific pathways, right, that have been established in the literature to have an impact in oncology or other indications, and sort of figuring out what are the molecular players on that pathway that are amenable to small molecules targeting. So I think there's kind of two different angles, depending on whether you're coming in from the chemistry side or whether you're coming in from the biology side. But at the end of the day, I think it all culminates on sort of what is the tool that you have at hand, right? So small molecule, is it oligo or even circular RNA? I think recently, which there's a new company that's been formed in that area, right? And what then makes sense, I think, for that platform in terms of targets. So that would be how I summarize that. Yeah.
Thank you. In that sense, the logic is yes, please.
If I could jump in, I just would say that, in all of this, I think the key question for all of us is therapeutic index. Can we safely dose a patient, treat the tumor, or whatever the disease state is without having untoward side effects. And related to that is, how do we identify those patients that are most likely to benefit from the therapeutics we're developing? So, the way we've approached that at Accent is we front-ended that question by investing quite a bit of time and energy in looking across the genome of RNA binding proteins for selective essentiality. What I mean by selective essentiality is situations where we genetically ablate the gene by using CRISPR or shRNA, and we look for situations where a certain set of cancer cells are sensitive to that knockout, but other cancer cells are not. And even within a specific cancer indication, we prefer situations where we can categorize responsive and non-responsive cells. We think that gives us the greatest opportunity for a therapeutic index, as well as a starting point for understanding the molecular or genetic basis of patient stratification. So all of the targets that we advance to drug discovery meet that criteria of selective essentiality. It's early days, but so far in the targets where we've looked, we've been able to recapitulate that selective essentiality with small molecules as we do with knockout.
Marvelous. Well, we're running out of time, and I want to do that future look that I always love. And so I'm going to ask each of you to keep it really, really short, but tell me what you think is the most exciting thing out on the horizon for targeting mRNA. And I'm going to start with David. So, go, David.
Well, I already said my one about making cold tumors hot with sensing double-stranded RNA, but I've got another one, and that is that I think that there's a real opportunity in identifying more RNA binding proteins. At the moment, they're big experiments. They're 300 million cells. There's a lot of money. There's a lot of huge amounts of mass spec that's required. I think the time is now right for new technologies to come in, new ways of purifying the actual substrate so that we can get down to something that laboratory people can work with. So instead of using 300 million cells, we're going to use 3 million cells. Instead of identifying 800 in a highly variable, 800 proteins in a highly variable setting, we're going to be able to identify a couple of thousand. And I think that's going to be a quantum shift, and that's going to bring mass spec to the fore, and we're really going to find what we need in the very near future.
Thank you. Jennifer, just a sentence or two on what you think is exciting out there in the future.
Well, I think that having a molecule which is orally available that in essence recapitulates what an antisense molecule would do or an RNAi molecule would do is pretty exciting in its own right. I actually think that sort of the next thing over the horizon are molecules that do something a little more interesting with the RNA. They actually elevate its translation or its lifetime or cause read-through in ways that are predictable and reliable. And therefore, it's not just a question of preventing the protein from existing, but actually having the right protein at higher levels. That would be a remarkable achievement. And it's really more like an agonist as opposed to an antagonist.
Right. Cool. Bob, do you want to add?
Well, I think there are some really exciting things that are happening in the community that are sort of orthogonal to the RNA part of the biology, but will intersect it. I think from a diagnostic perspective, circulating cancer cells are really exciting, and the ability to sequence circulating cancer cells, I think, has tremendous possibilities for diagnostics. I think the intersection of monotherapies based on RNA modification, but also the intersection with immuno-oncology that David was talking about, we're very excited about at Accent, clearly. And then finally, I think that, as we are studying these systems more and more, the concept of point point mutations as drivers of oncology has been very important, but I think a lot of the low-hanging fruit has been picked there. And I think that the concept of synthetic lethality and understanding those connections is going to be more and more important for the future of oncology therapies.
Thank you. Carolyn.
Yeah, I would keep my very succinct and short. I'm actually just waiting with bated breath for all these for companies like Arrakis, Accent to have our molecules in a clinic and sort of see how that's going to deliver benefits to patient. So I think it's a long time coming now. We've been doing so much work in this space, and I'll be really excited to sort of see how that pan out when we get to treat patients and deliver impact to them with these new molecules.
Thank you. And last, Joan?
Yeah, well, in a nutshell, I would just like to say, based on everything I've heard today, that it seems like there's a real prospect of personalized medicine emerging based on targeting individual mRNAs, and I think that's incredibly exciting.
Wonderful. Well, thank you all. Sarah, back over to you.
Wonderful. What a great discussion. And Carolyn, I wait with bated breath, as I'm sure everybody else in our audience does as well, for the developments coming out of Arrakis and Ascent. So thank you all. Thank you, Joan, Bob, Carolyn, Jennifer, and David for this great discussion. And Linda, thank you for a great moderating job. And your job is still not finished yet. So this ends the formal portion of the virtual salon. So for those of you who have to leave, please know that you can do so. When you leave, you will be asked to take a very quick survey. Please do so. If you would like to ask a question, please remember that you can put it in the Ask a Question box. But for now, I'm going to turn it back over to Linda because we have a number of questions that have come in, and Linda will lead this portion of the discussion.
Thank you, Sarah. Yeah, we do have a number of questions. David, do you think that deal terms are already set for the space, regardless of platform validation?
Yeah, that's a great question. So it depends on the stage of the asset, I think. I think nothing speaks like clinical data, but of course I don't think we're there yet. So there's a lot of estimations one has to make about what the risks are of the project not reaching milestones that are being set. But I think that there's a real appetite for risk in this space, and I think people, if they get the science and they get the possibility and the fact that you probably are going to have a first-in-class sort of molecule, I think there's a lot of upside for that. So I think, yeah, the deal terms aren't set, but I think, well, I guess we're just looking at what deals are being done and...
Yeah.
Sorry?
Big bucks and big excitement, yes. Sorry about my... Well,
yes. And if in other areas that have had this sort of explosion of interest, such as immuno-oncology, I think that there's a lot of interest in these because it is such a new area and has such great potential. But when that pans out, I guess all this will solidify and we'll really get a good understanding once things get into the clinic.
So another question is addressing off-target effects. Do you think targeting mRNA has any greater off-target liability than other approaches? Who wants to take that one?
Well, yeah, I could have a swing at that. I'm sure everyone's got an opinion on that one because, I mean, that is arguably one of the questions that we all get asked. I've spoken to dozens of biotech and pharma companies, and that is possibly the question we get a lot, which is, if we go after a specific pathway, like some big molecular machine that involved in RNA processing or splicing, what is going to happen? Are all mRNAs going to be affected equally? Are we going to see effects in different cells over a temporal fashion, that sort of thing? So there's lots to answer there. But I guess, we see some data on this that it may not necessarily be too much of a problem. A good example would be Exonate's splicing kinase inhibitor of SRPK1, which they are using as a way of modulating VEGF expression in back of the eye diseases. You would think that by modulating SRPK1 phosphorylation of splicing machinery that you would get a global effect. And yet, at least in their animal models, you can, and it's progressing to the clinic, that there's limited off-target, well, if there are off-target effects, that they have limited effects in terms of biology. Biology and toxicology, and of course the clinical trials tell us what's happening in people, but at least the indication, I guess, from their progress towards the clinic is that that's not necessarily a bad thing. You've got a global inhibitor, it's hitting lots of different RNAs, but the actual impact on disease isn't necessarily too bad. So I think there's a lot of hope there.
Carolyn, what about delivery? Certainly that for oligos and mRNA has been such a huge challenge. Do you see that as advancing rapidly?
Yeah, I think we're definitely seeing some progress there. And as I've said, I think there are companies now that's moving sort of outside of liver to target other tissues like muscles. But we're still, I think we're very far from the holy grail of being able to sort of do that in a wide fashion to target any target tissue of interest. So that is probably going to take some time away. But I also noted, as I mentioned earlier, I think the circular RNA company that recently just got formed by our friends at NPM, Orna Therapeutics, where they're using a different type of structure for RNA, and potentially that could enhance, with a circular shape, we'll be able to enhance stability and perhaps overcome some of the packing issues that we see with viral vectors. I think that is a very creative and orthogonal way of solving the delivery issue by using a different type of RNA in a geometric sense. So I think there are so many interesting ideas out there that might be a bit different from what we sort of typically think about delivery, which is just, are we using, nanoparticles, or are we using LNPs, or viral vectors? Right? But there are, I think, other approaches that could potentially even sort of solve the problem in a different way.
Right. Jennifer and Bob, you're using small molecules, so delivery is less of a challenge, but one of the audience asked about brain penetration. Are you targeting RNA in the central nervous system?
Well, I can speak for us in that there are targets that are of interest to us, which are in the central nervous system. So yes, we need to get into the brain. For small molecules, getting into the brain isn't automatic. I mean, the SAR can be a little challenging to solve that problem, but there are plenty of people who solve that problem. And so I think that we have good prospects for solving that.
Thank you. Bob?
Yeah, likewise. I think that, again, because we are targeting enzyme rather than the RNA per se, a lot of the learnings that have been developed over decades are things that we can apply. Right now, we are not targeting exclusively diseases of the central nervous system. We're going after oncology more generally. But there certainly are clear indications that RNA modifying enzymes are involved in diseases beyond oncology. So for right now, we're focusing on oncology in general, but down the road, we could think about diseases of cognition, psychiatric diseases, things of that sort, where there are implications for RNA modifying enzymes, but things that are more in a embryonic stage of development.
Thank you. Well, I think we've probably used up our time, but it's been a marvelous session, and I tremendously thank the panel for their insights and for educating me and the rest of us. And thank you very much to the WEHI for sponsoring this, and to Sarah, Demy-Colton, and crew for hosting it.
And Linda, we do want to thank you for bringing WEHI to us. It's a wonderful institution. I've traveled there, been there many times in the past, and so I'm very pleased that we were able to do this today. I do want to thank WEHI for being a diamond sponsor of the Virtual Salon Series. That's really very important for us all, and for bringing us this great discussion today. We'd like to thank the audience as well for joining us. And as I noted earlier, if you're still with us, please do take that brief survey as you leave. And please join us for our next Demy-Colton Virtual Salon, a critical healthcare burden needing rapid diagnostics to improve patient outcomes, sepsis. Until then, everyone, stay safe. Stay safe. Signing off.
Thank you.
Thank you.
Bye now.
Bye. Thank you.